First of all, thanks for inviting us to present our paper. And just to quickly introduce myself, I'm Adalberto Claudio Quiros. I'm a PhD student at the University of Glasgow, Computing Science Department, and I'm supervised by Yuan, who is also attending the symposium here. And I first wanted to start with the motivation of why we decided to work on this model. So we are firstly motivated on how to learn morphological patterns in histology. So how we can pick up tissue representations solely from the information contained in the image. And this is in an effort to try to overcome that clinical labels are expensive in time and effort. And also, once we are able to gather these histomorphological patterns, we can correlate them with clinical annotations to study these phenotype, the relation between these phenotypes and the annotations. So far we have done it in two different ways. The first one is using generative models like GAN in here in what we are going to present next. And in the other approach is just by using self-supervised learning. So just to jump into the GAN model, we started with a big GAN. And all the features that that model has, like the spectral normalization, self-attention layer, the orthogonal initialization and regularization, and also the hinge loss. And we decided to replace the hinge loss with the relativistic average discriminator. So the motivation there, I'll give you some examples. It was just an empirical study. What we have seen so far is that this loss, in general, stabilizes the model more and give us better results. So after that, we were able to achieve images with high quality. But like I said in the motivation, we wanted to learn representations that actually contain like cell characteristics found in the tissue. So we introduced, based on the style GAN, we wanted to introduce some of the features because from the paper itself, it seemed that the noise vectors had some representation learning. So we introduced three features from the style GAN, the mapping network. What that network allows is to modify the original latent space from an isotropic Gaussian into an alternative one. And then we also use the adaptive instance normalization. So that's just a kind of layer that does batch normalization after its convolutional layer, and then it scales the values based on the input noise vector for each of the instances, so each of the images that you're going to generate. And we also use style mixing regularization in order to further localize the features in the latent space. So the way that you do that is that you take two different latent vectors, you pass them through the mapping network, and then you randomly choose a cutoff layer on the generator, and from that point upwards, you feed one input latent vector, and the cutoff to the bottom, the other latent vector. So, like I mentioned before, we replaced the loss of the original big GAN, the hinge loss, with the relativistic average discriminator, and this is kind of the motivation behind it. So, as you can see on the left, we have the relativistic average discriminator, and on the right, the hinge loss. And these images are mid-training of the GAN model. You can see here that there's a collapse started to happen, and then you get some batch effect in which you see images that look like each other a lot, and there's just very few variations. So my personal hypothesis behind this is that hinge loss, you can see it as a the reverse KL divergence between the generated distribution and the real one. So it doesn't reproduce the data as well as the original GAN loss, which is the Jensen-Shannon. And the good thing about the relativistic average discriminator is that it doesn't label each image by real or fake. It just compares a batch of real images with the fake generated one, and then the discriminator just to try to establish, based on the difference between the two of them, which one is more realistic in that sense. So just to continue with the image quality or image generation here, on the left, I have two panels for two different data sets. The left one is breast cancer data set, and on the right one, you can see the colorectal cancer. And then in both of them, the left column here, these are generated images, and on the right, you see real images. So what we have done here is take the generated and real images, and we pass them through an inception B1 convolutional neural network. And then in the feature space, we started to look the real nearest neighbors to the generated samples. And you can see that the real and generated samples in that feature space, they look a lot, they look quite similar in general. Another way that we try to compare the original distribution and the distribution of the generated samples is through the Frechet inception distance, which is a common GAN metric. So the way that it works is that this is just a measure of distance between two multivariate Gaussians. And then you will have the two, for the real image distribution and for the generated distribution, you will fit both of them multivariate Gaussians, right? So you can do it in different ways. You can use different feature space. That's what I was trying to say. So the first way that you can do it is just by using the convolutional features from the inception V1. So you can take 10,000 different samples for the generated and the real distributions, pass them through the convolutional neural network, and then for each of the images, you will have a 2048 dimension vector, and you can fit all that 10,000 into the Gaussian distribution, and then just compare the distance between the two of them. So that would be the normal way of doing the FID score. We thought that it would be also beneficial to get some kind of measure if we were to quantify the cell activity in the tissue tile. So we use a framework called CRImage, which is just an SVM tool that segments out the cells, and then it gives you a score for the number of cancer cells, the number of lymphocytes and stroma, and the cancer cell density. So in a way, it will also give you another vector for each image, and then you can fit that again into the multivariate Gaussian. And you can see here that we are reporting those values, first for the convolutional FID, the inception, and then just by using the vector representation from CRImage. And I mean, by now, these values are not that low, but you know, in the GAN world, I think they were fairly decent by the moment. The next thing that we did was to try to, in a way, quantify how real the images were. And we tried to do that by creating some kind of test for the pathologists. So what I'm showing here is just 10 different samples that we gave pathologists to annotate between real and fake. And we asked them to quantify them from 1 to 5, as 5 being the most real. I believe we gave them like 50 samples for them to annotate each of the pathologists. This will be an example for breast cancer, and this is another one for colorectal cancer. And in the end, once you have that kind of a score, you can try to put together an ROC curve with the classification performance. Something that I forgot to mention here actually is that the way that we picked out samples were, one, just by taking the 3 closest near neighbors from real images, and then a generated image. And the other one was by hand selecting. The motivation there was that some images are really easy to replicate, like lymphocytes. Images with a lot of lymphocytes are just, they tend to be like dense tissue tiles with these black dots. So those tend to be closer to the real samples. And well, the performance you can see here that it's close to random classification. And I think this is the most interesting thing from the model itself, which is the representation learning part. So that alternative layering space after the mapping network. So in this case, what we did was to generate 10,000 samples images. And then with each of the latent vectors that generate the image, you can do a UMAP dimensionality reduction. And then for the generated image, you can try to find a label to see if there's any kind of a structure in that latent space. So for the breast cancer data set, what we did was to use again the CR image. And then we created 8 different classes accounting for the cancer cell density in the image. And then we just label each of the latent vectors. And you can see here that going from this point here, and if you take a diagonal going to the right, it increases the number of cancer cells. And you can visually actually see that from this point until you get to most cancerous tiles in here, like densely packed. And then for the colorectal cancer, we took advantage of those nearest neighbors between the generated images and the real ones. So in that case, we took 10 nearest neighbors, and then just by majority vote of the kind of class that was closer to the generated image, we assigned the tissue type. And again, you can see around here that depending on the region, you will generate a different kind of image, more lymphocytes if you are around here. If you go down to the right, tumor tissue type, down stroma, and then adipose and some background. So in a way, we tried to also see what was the effect of introducing those features from StyleGAN. And in the case of the breast cancer data set and those 8 different classes accounting for the cancer cell density in the tile that I mentioned, what I'm trying to show in here is what a model like the big GAN without those features will show in the layering space as compared when you introduce them. So you can see, especially in the bottom part, these are randomly placed vectors in the latent space, depending on the number of cancer cells. But when you add those features, they tend to constrain themselves to particular regions of that latent space. And this is another example for the colorectal cancer tissue. I think this one is even more clear here. So another thing that we tried to do was linear vector interpolations between 2 points and see the effect of the generated images as you travel along with the latent space. So I'm showing 2 cases here, one for the colorectal on the left and another one for the breast cancer on the right. And in this case, we started off with a stroma sample and then ending up on a densely packed lymphocyte tile. So as you keep moving along the latent space, you can see the number of cells keeps changing. At some point in here, you have a densely packed tumorous tissue, and then you travel along and end up with a lymphocyte tile. And in the breast cancer, a similar case in here, but going backwards from a cancerous tissue to more lymphocyte. Now, similar to what we did with that ordering of the latent space, I wanted to show here what is the difference in effect with having those features from StyleGAN and not having them. So A and D include those features, and B and D are the big GAN model. And you can see from B and D that there's some kind of fading in the image when you try to do that linear interpolation in both cases. Whereas, for example, here, with those representation learning properties, the number of images in the tile seems to be absolute. It doesn't seem to have that fading effect. Now, the other thing that we show is that you can do very simple linear vector operations that translate into features in the image. So just to give you 2 examples here, again, for the two different kinds of cancer tissue. On the right, you see a simple addition of features for lymphocytes and tumor tissue. And then down below here, I'm showing you the original vectors and the vector after the transformation as compared to the entire population of all the generated samples that we had before. And in this case, another vector operation in which we just took the vectors from two tumor tissues, subtract them, and then add some stroma, and we get some stroma-like generated tissue. And again, just showing, kind of trying to give an intuition where those vectors will move by doing those operations. So like I said before, the motivation was to try to find representations of tissue tiles. And this is more a spin-off that we did after the paper, that I think it's where it brings in how we can use real data with the model. And the limitation from the original PathologyGAN was that we couldn't find representations of real images in a quick and easy way. So we just introduced an encoder that it will train along with the GAN itself, but it wouldn't interfere in the GAN learning process, and it will map back images to the latent space. So the way that this encoder is trained is just by a simple mean square error of the original latent vector that generated an image, and when you map it back to the latent space. So the reconstruction of the latent vector. And now the question would be how useful these latent vectors are. Can we use them for different tasks? So the first thing we did was just to check real images in that latent space to see if there's still that structure according to the properties of the tissue. And similarly to the image that I showed before, we generated image, we have the same one, but for real images, the projections on the latent space. And you can see that there's still a structure in that latent space. So in here, you will have outwards more densely packed cancer cells as you inwards, there's less cancer cells in the tile. And then with respect to the colorectal case, its real image had an annotation from a pathologist telling the tissue type. And you can see that depending on the region, again, the tissue type is constrained in the latent space. Another thing that you can do is just take real samples and map them to the latent space and then reconstruct the image with the generator of the GAN. And in here, rather than to show that there's a one-to-one match in the pixel, we wanted to show that the actual tissue architecture is similar when you, as compared between the reconstruction and the original image. So at the top, you have the real images, and at the bottom, the reconstructions. So for example, in this case, you can see that both of them show to have a high number of cancer cells as compared to here, that it's mainly composed by just tissue without any kind of cells, like comparing the reconstruction here. And the same thing in here for the stroma in the case of the colorectal tumor or the mucus, for example. So the other, there's two other tasks that we tried here, and this is what I think it gets more and more interesting on how you can use these representations of the tissue for different applications. So like I mentioned before, the real images had annotations from a pathologist telling the tissue type, right? So in this case, we wanted to see if we could train a linear classifier over the representations of the tiles and then report the classification with this logistic regression. And as you can see here, the AUC, it's comparable to other supervised methods. Let me see if I say this right. So in the end, although there's a loss on performance when you have the completely supervised methods, especially compared to the Bayesian DNN, the fact that you are training these representations in an unsupervised manner show that you are picking up cellular features. And then another application that we did to use these representations will be just to put them into the context of whole slide images. So you break down the whole slide image into the tiles, find the representations for the tiles, and then in the lung cancer context, try to use multiple instance learning to predict if there's tumor presence in the whole slide. So we compare this performance to another completely supervised method from Roderick et al. Using an Inception v3. And again, the AUC in this case, it's comparable and the accuracy is within the ballpark. Now, there are a few things that I think there's room for improvement in the model. I think the first one is to do a further exploration on the disentanglement of that latent space and clinical features. Like I think there's been a few GAN papers already trying to break down which dimension, if you can find alternative latent space from the original style GAN that will transform accordingly with a label that you want to give, and maybe bringing that into the model will give a better split between a meaningful separation of the features in the image. Then I think one of the things that this model is prone to is to be affected by imaging process, like the variability between staining and zoom from different institutions. I think you can see that especially in this image here, where UMAT is probably enhancing it, but there's clustering between some representations that probably is because they are picking up some nuances from the image. And then just trying to build a model that is able to handle larger resolutions or being more stable. So in this case, I'm just showing generated images for 448 by 448 at the top and then real images at the bottom. And I think this is where the model starts to struggle. So going further than this will be another way to build on or improve on what we have already. Now, I just wanted to wrap up because we get this question a lot of, so you always say that you are motivated of learning this tissue phenotype. So what's interesting about that? So especially from the last applications, you can see that we can build unsupervised representations of the tissue, but what it's interesting is that you just pick up information from the tile itself, and then you try to correlate it with other clinical annotations just to find the representation from the tissue itself rather than the correlation with the clinical annotation in an end-to-end solution. And an alternative to what we show here is the self-supervised learning, as I mentioned before. I also wanted to acknowledge and thank some of the people that, well, some of our collaborators that we work with at the New York University, other people from computer science, and then the Bitson Institute and the University of Glasgow. And thank you once again for inviting us for this presentation. And I also wanted to point out that through the process of trying to publish the MELBA paper, I think the reviews were constructive and it made our paper better. And I think it was a nice experience, and I wanted to mention that because I think it's worth saying. Cool. So yeah, that was it.